In this series of figures, we're going to look at the fluid compartment separations just a little bit more closely. In A, at the top here, we have the separation between the ICF, the intracellular fluid, and the ISF, or the extracellular fluid that surrounds every cell. Down here in B, we have the capillary wall. The capillary wall separates the plasma on the inside of blood vessels from the interstitial fluid found on the outside of those blood vessels, again, surrounding the cells. And of course, every cell is surrounded by a cell membrane, and so we can take a sampling from here, or the epithelial cells that form capillaries, or even a red blood cell, and take a look at that membrane, and it's all going to look very similar to what we have up here at the top in A. What we find is that we have a phospholipid bilayer as our composite scaffolding baseline structure for a membrane, but then we have these associated structures with the lipid bilayer that add functionality to it. We have the glycocalyx, that complex of proteoglycans and carbohydrate groups that sort of project out into the interstitial space, as well as membrane proteins that are associated with the lipid bilayer. And these can serve a whole multitude of functions, such as enzymes or receptors or transport proteins or channels, as we see here, an aquaporin channel for water. Now, the key characteristic in terms of permeability of the lipid bilayer is that it is semi-permeable. It is basically permeable to only what is allowed to go through it via transport proteins and channels for hydrophilic solutes. Hydrophobic or fat-soluble solutes, if they are very small, will readily pass through. In other words, things like oxygen will readily pass through this membrane without requiring a transport protein or channel of some sort. Despite the fact that it is lipophilic, it would be repelled by the phosphate heads here, but it can actually fit between them and get into this sort of fatty middle layer where the fatty acid tails of our phospholipids are found. There it is not repelled. And so because it can get between the phosphate heads, and then it's happy to be in the inner layer here with the fatty acid, the hydrophobic core, if you will, of the membrane, that can readily pass through. Carbon dioxide is the same way. It can pass through the lipid bilayer unaided by anything other than a gradient. For larger fatty substances, they would be repelled by these phosphate groups, and those actually require a carrier. And so large fatty substances or polar substances generally require some sort of transport mechanism, a protein, to aid their transport across the membrane. If we come down here to the capillary wall, what we find is that transit across that barrier, it could occur across the membranes of these epithelial cells. It could occur across the apical layer and then the basolateral layer, right? And something could pass out of the bloodstream and into the ISF or vice versa through the cells. And of course, that would be common for a small lipophilic substance like oxygen or carbon dioxide. But our larger substances, our polar substances, tend to pass through these paracellular clefts. There are little gaps between the epithelial cells that are much bigger and allow our solutes to pass back and forth readily. Now, small solutes, ions, nutrients, waste. Large solutes like proteins are not readily permeable across most capillary walls.